Good afternoon, friends, colleagues. It's a great pleasure for me to be at the ULF again and to speak with you on Vanishing White Matter. And it is really loud, the music. Say it again?
It's not too bad for us.
Okay. What I'll start with is with an overview of an introduction on Vanishing White Matter, because every year there are new people and I want everybody to be on the same page and understand what's going on. And that means that for some of you, it's something that you've seen before and already heard before. And then so it might be a little bit repetitious, but that's for the people who are here for the first time. So this is my research funding, because there's no conflict of interest. We have a patent on therapeutic effects of guanabans in Vanishing White Matter in the USA and in the EU. We have an orphan drug designation. I'm a consultant for Calico on therapy development for Vanishing White Matter and a co-investigator for the Alexander disease trial of Ionis. So, Vanishing White Matter comes with different phases, and the phases depend on the age of onset. This is classic Vanishing White Matter. Classic means that it's the most common form of the disease. So most commonly, the onset is between 2 and 6 years, and it's a disease dominated by motor decline. So mostly cerebellar ataxia and less obvious cognitive decline and sometimes epilepsy, but typically epilepsy is not the biggest problem. On top of the slow chronic decline, there are episodes of major decline provoked by stressors. So small infections, fever, minor head trauma may provoke a sort of acute decline with irritability, lowered consciousness, and also loss of motor skills, followed by typically partial or complete recovery, but sometimes patients die in the course of a decline, and they may die after variable number of years to decades. I'm going to show you. Then there's early onset Vanishing White Matter. The onset is before birth or in the first year of life, and then it's a generalized encephalopathy with very little or no development, fast regression, severe epilepsy, irritability, and these patients die very soon. So this is a very rare form of the disease, but it's devastating. Then late onset Vanishing White Matter has its onset in adulthood, can have an onset in the 50s, in the 60s, in the 70s, and then it's characterized mostly by cognitive decline and behavioral changes, and later and less striking motor decline. It's a very slow disease with a variable disease progression. The stress-provoked episodes are less prominent, and they live for a very long time. So clearly, disease of onset is a predictor of the disease course. Both what signs are predominantly present and the rate of progression. So we did a natural disease study that was done by Eline Hamilton. We have a registry since 2004, so it's 20 years old. And we have currently data on 420 DNA-confirmed patients worldwide. And we collect data, clinical questionnaire, and quality of life and handicap questionnaires, and follow the patients over time. In the Netherlands, we have centralized diagnostics, and that means that we pretty well know what the incidence and prevalence of the disease is. So the incidence in the Netherlands is approximately 1 in 100,000, and the prevalence is 1.3 in a million inhabitants. And I think it's similar. We have no reason to think it's different elsewhere. And that means that there are many patients undiagnosed. Then this is what we call Kaplan-Meier plots, and we divided the patients according to age of onset. So black is younger than onset below the 1 year of age, red between 1 and 2, yellow between 2 and 4, purple between 4 and 8, blue 8 till 18, and green above the age of 18. Now this is the age. And then this is the proportion of patients surviving. This is ambulation and this is cognition. So what you see is the later the onset, the longer a function is preserved and the longer patients live. The thing is, if you do it differently and you look at disease duration, you get an impression of the rate of progression. And then you see that the very early onset disease is extremely rapid. And then as soon as the onset is a bit later, you can see it spreading out. And then from the age of onset of 4 to 6 on, you can see that the rate of progression remains the same. This is true for ambulation, for survival, but not for cognition. You can see that the cognitive decline in adults is relatively early and fast. These Kaplan-Meier plots look pretty like you can predict the disease course. You can plot your child on the Kaplan-Meier curve and you can see where it ends. This is not true at all because that's just the mean disease course. Here are the individual patients. So you can see that the decline in the very early onset cases is really very rapid, but that as soon as the onset is later, it totally spreads out. Like onset between 2 and 4, you can see that some patients die very soon and some patients still live in their 30s. So that's extremely variable. And then you might think, but still the lines are straight. And that means that for one patient it's still predictable, but this is not true at all. Because that's just because of scarcity of assessment points. Because if you have multiple assessment points, you can see that the line is going up and down with sudden declines. And here also a patient suddenly dying. So all in all, vanishing white matter is an extremely unpredictable disease, which makes it very difficult. Then patient numbers. This is age of onset and then number of patients. So you can see that 2/3 of the patients have an onset below the age of 6. But I think that there's something to say to it. I think there's underdiagnosis, major underdiagnosis here. So that this plot is not correct. And the second thing is, if you look at patients that are living now, 2/3 has an age of 16 years or older. That means that because some people say, but the biggest unmet medical need is in the very young patients. I don't know that, because these patients live a very long time. And there's also a big unmet medical need. So that's something to think about. Pathology. This is a whole brain slice. Pink is the cortex. The cortex looks fine. Then within the cortex is the white matter, and you can see here the big hole. That's what vanishing white matter is about. The white matter vanishes. And here's the big hole. Here you can see that it's not a big hole, but the myelin is lacking. You can see that here's the myelin, so this should be dark purple. It's lacking, and in the central part of the brain it's present. Then looking under the microscope, it's mostly what we call the glia that are affected. Oligodendrocytes are the glia that make myelin, and they are normally looking like this, and in vanishing white matter, they may look foamy. They also are immature, and they don't do what they normally need to do. They need to make sufficient amount of myelin, and as you can see here, they don't do that. Then astrocytes are the housekeeping cells that are there for homeostasis in the brain, and they keep everybody happy and healthy. And these are normal astrocytes. As you can see, little beautiful stars, and in vanishing white matter they look really ugly. They have a few stunted processes, and they look really very abnormal. Then if you stain them for immaturity markers, normally they are negative for nestin. Nestin means that they are not mature, but in vanishing white matter you can see that they are positive for nestin. That means that also the astrocytes are immature. Astrocytes are also for making scar tissue. As you can see, they don't do that. They actually do very little in vanishing white matter. Then Bergman glia are specific astrocytes in the cerebellum, and they are normally located here. We call this the Purkinje cell layer. In vanishing white matter, they are mislocalized. They are in what we call the molecular layer, and they again look ugly. You may think, why is he showing this detailed pathology to us? I do that because I'm going to show therapy effects to you, and I want you to realize that what we show is real. Vanishing white matter is caused by a defect in a factor EIF2B. And this vector EIF2B is composed of 5 subunits: alpha, beta, gamma, delta, and epsilon, each encoded by a separate gene, EIF2B1, 2, 3, 4, 5. And this vector is conditional for the translation of DNA into a protein. Now, protein is about everything. If this vector would be, completely defect and do nothing, there would be no protein production, so there would be no life. You cannot live without EIF2B. So patients have small mutations, they need remaining activity of EIF2B, and otherwise you don't see the life. EIF2B does not only that, but also regulates the rate of protein synthesis. It regulates how much protein you make at a time. It's very active, then lots of protein is produced. If it's not that very active, then the protein synthesis is down. And this is something critical because under stress conditions, protein synthesis needs to be downregulated. And for instance, in circumstances of growth, protein synthesis needs to be upregulated. So this is something that's regulated all the time, and that's done by EIF2B. I'm going to show you the stress response. It's very difficult, but if you follow me, it's doable. Here you can see the stressors. And what's important with stress, I don't mean my mother is disciplining me, or I don't want a venipuncture. I hate the doctor. Those are stresses, but not the stresses I mean. The stresses are physical stresses, like fever, head trauma, viral infection, and things like that. So here are the stressors. Reactive oxygen species, viral infection, heat, amino acid starvation, misfolding of proteins in the endoplasmic reticulum. They activate different enzymes, and all these enzymes do the same thing. They phosphorylate another factor, EIF2, that then tightly binds EIF2B and inactivates it. That's what stresses do. That's how stresses come to decrease EIF2B activity. And then subsequently, you see here, general protein synthesis is down, and the ATF4 transcription response is activated. And the ATF4 transcription response is critical for the cell to help decide, is it going to be repaired? Is the cell going into apoptosis? Is the cell going to die? So the ATF4 transcription response is pro-repair. Unless the stress is too much, then the cell will go into apoptosis. There's also a feedback loop, the GAT34 feedback loop, because of course the stress is also stopping, and then everything needs to return to normal, and the GAT34 feedback loop does that at the level of EIF2. Now, in vanishing white matter, EIF2B activity is always down. So that means that this response could always be activated. Now, we've never been able to find that general protein synthesis is decreased or deregulated, and I think the remaining activity of EIF2B is sufficient to maintain general protein synthesis, but the ATF4 transcription response is always on. And also, including the GAT34 feedback loop is on. And that means that we find that EIF2B phosphorylated form is down. We think that this is critical in the disease, that the downstream part of what we call the integrated stress response is always on. Now, so far the difficult part. We have, of course, to think of treatment. And what the good thing about vanishing white matter disease is that it's in a pathway in which you can think about treatment. So first of all, avoid the stressors, because that's clearly clinically deregulating the disease. And I think you all heard about that. Avoid fever, avoid infections, avoid head trauma. And I would say that helps to avoid the episodes, but the chronic decline is still there. You could also think of using drugs to combat the stressors upstream. The second thing you could think of is stimulate EIF2B activity directly, that's here. Modulate the feedback loop through CAD34. Reduce components of the ATF4 transcription response, that's somewhere here. Or modulate further downstream effects. So, what we did is we made vanishing white matter mice. So we took mutations that are very severe in humans, and we gave the mice the same mutations, and these mice have a phenotype. I'm going to try whether I can... Yeah, oh. You can see the mouse is extremely ataxic. This is the double homozygous mutant, so it's really very, very, very pitiful. I should stop it. I don't like to look at it. But you can see that the mouse has a severe ataxia. It's also smaller, it has epileptic seizures, it has a shortened lifespan depending on how mutant it is. So we have 2 mutants, so we can also crossbreed them. This is the 2B4 homozygous mutant, this is the 2B5 homozygous mutant, this is the 1.5 mutant, and this is the double homozygous mutant. So we see we have a whole range of disease severities, just like vanishing white matter patients come in different disease severities. We have mice with different disease severities. They also have brain pathology very similar to the humans. I'm going to show you. So myelin is down. What you can see here, mouse astrocytes look a little bit different from the human astrocytes. But in vanishing white matter, they still look ugly and they're nestin positive. So they look ugly and they're immature. Then the Bergman glia are normally localized also in mice, in the Purkinje cell layer, and in vanishing white matter mice. They're mislocalized, they're thick, and they're immature. So this is just the same as what we see in humans. Now, EIF2B activators. They're chemical compounds stabilizing EIF2B and directly increase EIF2B activity. The first compound has been found by Carmela Sidrowski, who now works with Cadicode. At the time, she worked for UCSF, I think. And so that is a chemical compound that was found by compound screening. So her group and our group both used an EIF2B activator to treat control and vanishing white matter mice. And what we found is that the ISR activation, the abnormal ISR activation is decreased, and that the motor performance and neuropathology are improved. What you see here, this is the 1.5 mutant mouse treated with placebo, and the yellow cells are astrocytes that are immature, and with the EIF2B activator, the immaturity is down. This is the untreated mouse, placebo-treated mouse. You can see the ugly Bergman glia. Here they are back in their normal position, and also there's more myelin. And this is the balance beam. That's much improved, but I'm going to show you the mice. I'm going to try to activate them all. So we have a catwalk, and this is a normal mouse walking in the catwalk. Of course, a mouse doesn't walk like that, but we slowed the movie a lot, but so that you can see how the movement goes. Then this is the vanishing white matter mouse, untreated. And you can see that it's very unstable, that is leaning against the wall. It uses its tail to stabilize, and so the mouse is ataxic. And this is the vanishing white matter mouse with an EIF2B activator. And you can see it's doing much better. So the conclusion is that EIF2B activators are highly promising in vanishing white matter. There's going to be a lecture on it later in the program. They have a good short-term safety. They're novel compounds, so the long-term safety, let's say over 10 years, is unknown. And the trial is in development and will be discussed. So then the second possibility. We'll discuss all possibilities. Quanabens is an alpha-2 adrenergic antihypertensive drug, FDA approved. It's very old. It was entering the market in the late 70s, and it has been safe over decades. It's no longer used as an antihypertensive because it's just replaced by more effective drugs. It also impacts the ISR through the CAD34 feedback loop, and it also ameliorates motor dysfunction, ISR activation, and neuropathology. in a dose-dependent manner. So what you see is the higher you dose the guanabans, the lower the AT4 transcriptome activation. Astrocyte becomes more mature. You can see these are the mislocalized Purkinje glia. They go back to where they belong, and myelin goes up, and the catwalk improves. But of course, the movies are much nicer to look at. So I have also movies for those. This is the wild-type mouse. Then this is the vanishing white matter mouse with placebo. And you can see it's much slower. Again, it leans against the wall. It's very ataxic. And this is the treated mouse. So that's also much better. So the conclusion is guanabans is highly promising in vanishing white matter, safe in long-term use, but there are alpha-2 adrenergic side effects. They have rapid tolerance, but still there are side effects. And there's a trial in progress. It's a phase 1-2 trial. In Amsterdam, it's an international monocenter trial. It's open label. We match with historical controls to avoid the placebo group. And we include patients with early onset disease. They still need to be able to walk with the most life support of one hand to avoid too much irreversible brain damage. Because if you start the treatment in a patient who is in an advanced, far advanced stage of the disease, you are unlikely to see the effect anymore and nobody gains. We are allowed a maximum of 40 patients in the trial, a minimum of 30. The disease onset is below 6, still able to walk, as I said. And we dose up until the maximum tolerated guanabans dose, because we know that there is dose-effect relationship. And we started in May 2021. We will include for another year, so May 2024 we'll stop including, and the whole trial will end at May 2025. We have, as I said in the beginning, a patent. We have an orphan designation, and if effective, we would like to go for registration and making the drug available at cost price. Modulate the AT4 transcriptome. So what that means that we try to reduce components of the AT4 transcriptome, because the question is which component of the AT4 transcriptome is now causing the disease. So we did that by making CHOP lower, GAT34 lower, or ATF4 lower, by crossbreeding them with other knockout mice. And we have lots of movies. This is the wild type, the vanishing white matter mouse, you already saw it. Then together with the CHOP knockout. You see it does better. This is the GAT34 knockout. Also does better. And this is the ATF4. And the ATF4 heterozygous, it's not possible to crossbreed with the homozygous because it's not viable. They don't live. So we did that heterozygous, and that has the least effect. ER chaperones, we did that. We used TUTCA or 4-BPA, and those had no beneficial effect. We also tested pridopidine to modulate further downstream effects and reduce mitochondrial dysfunction. And that had a very small effect. So on most parameters were unchanged, and a subtle beneficial effect on some parameters. So the conclusion is that eIF2B activators and guanabans have the greatest beneficial effects in vanishing white matter mice, which are not humans. This means that we are thinking of testing the therapeutic effects in trials, and we have now very small, an ultra-rare disease with a very small disease population and multiple drugs. That means that we have to think about trials. An ultra-rare disease may mean lack of patients, and then the trial will fail. A highly variable disease course. So small and short trials are unlikely to be informative. Soon irreversible brain damage, then we'll see nothing anymore. So we must aim for rather early in the disease course. And different phenotype for early and late onset means that we must stratify for age of onset. You cannot just make one big trial that also will not be revealing. So what we did to help guide trial development, we made a consortium of clinical experts on vanishing white matter. So this is Josh Bonkowski. He will also give a talk. Ali Fatemi from Kennedy Krieger Institute, Adeline van der Ver from Philadelphia, Brent Vogel from UCLA, Ingeborg Kregelo-Mann from Tübingen, Enrico Bertini from Rome. I think Enrico is here. He was here. Geneviève Bernard from Montreal. And Nicole Wolf from Amsterdam, and she's here. Our aim is mostly to help govern trial development. First of all, trial setup. What we feel is all therapies should be assessed in adequate trials before application in patients. You know, we had this very painful experience with Lorenzo's oil in XALD, and that led to decades of misery. So we want to avoid that. And so all therapies should be properly tested in adequate trials. And we wrote a consortium paper on trial setup in vanishing white matter, which was published in 2022. Then we also wrote a core protocol. We want a paper on the core protocol. We would like the trials to share the core protocol to test multiple drugs in parallel in studies that are adequately powered. And also, if they share the core protocol, the placebos can be shared so that patients on placebo can be minimized and we can maximize the patients on the therapy to be tested. So that's the primary aim of the consortium. We work with the Industry Alliance Office, and Priska is sitting right here. She's also here to help us. She supports the consortium and facilitates the interface between academia and industry. So I think it's my great pleasure to also let you know that the first industry trial will follow the core protocol, of which I'm extremely proud. Then we have the database in Amsterdam that we would like to transform into an international vanishing white matter registry. It's based in Amsterdam, but governance is by the Vanishing White Matter Consortium. It uses a cloud-based secure system that can capture and integrate data from any source. We work on following the guidelines of FDA and EMA, and right now we are reconsenting families to allow international sharing of data with other researchers in the industry, because this registry should be for everybody. And that's what we want to do. The registry needs to be suitable for natural history studies and other studies like the impact of the disease study, and Romy van Voorst will talk about that, and for providing historical control information that we may need for trials and for sharing results of trials following the core protocol. And that means that input from patients and families is crucial. I know it's painful for families to fill in all those questionnaires. You might hate it, because it also puts you on the spot on what you've lost, but we cannot do it. And we can make the structure, but we cannot fill in all the questionnaires and provide the information. So here's also a collaboration with patients and patient advocates. So I'm also very proud to let you know that we allied with the Vanishing White Matter Families Foundation in our papers on trial development and also on the execution of the trial. Patients and families need to participate in advisory and monitoring boards as needed, so they are part of they will also participate in the Vanishing White Matter Registry. They are on the board of the Guanabens trial. I hope they are involved in the EIF2B activator trial, and that they are active participants in research and in trial readiness. This is my final slide. It's the Vanishing White Matter team in Amsterdam Leukodystrophy Center. And here you can see the people who execute the trial. And I would like to point out some important people. Renate Verbeek is a pediatric neurologist. She's the trial sub-I. Marije Voormans is the trial coordinator. She's amazing. Claire de Koning is the main trial nurse. Daphne Schoenmakers helps us with getting the Vanishing White Matter registry ready and helps write the paper on the trial core protocol. Romy van Voorst is here and she's working on the Vanishing White Matter registry and also working on the impact of disease study. Menno Stellingwerf is our WIS-kid who works in R and does all smart things with statistics. So that if he writes a script, then everything goes by itself and it becomes something. And so that's very smart. And this is Nicole, who's also pivotal in the trial. And these are some people that, like the pharmacy and from the lab. And I thank you for your attention. How are we doing time-wise? I think we are a little short.
5 minutes beyond, but it's okay.
Okay. Anybody with a burning question? I'll be around anyway till tomorrow noon. And I'm ready to sit with everybody and answer questions. So, because some questions may be a little bit difficult for the public. So I'm happy to sit with everybody. Yes.
How long is the trial, and if you choose, can you get out of the trial?
That depends. I can give two answers. The Guanabens trial has a fixed end. So if you enter now, it's still 2 years. And what we made sure is that we provide the Guanabens for the participants also after the trial is closed. So we continue to provide Guanabens as long as needed. And I can answer the trial core protocol says that that trials need to be 2 years for each patient. Yes?
For the VWM trial, sorry. You had mentioned that if there's too much damage, it's not effective. Is that just currently in the trial or going forward?
It's general. If it's the blame is on the brain. If you have brain damage, then and you make a new MRI in 2 years, it will look the same. So the brain is very bad in repair, and that means that if something is already damaged beyond repair, that it will not be repaired, even if you give Guanabens or an EIF2B activator.
Will there be standards placed once you have the trial or the 4 years is up? Will you have standards in place that will classify if there's too much damage in place, or
we have it for
to indicate that?
We have it for the trial. I think after the trial, the whole situation will change. Let's say I'm going to picture a very optimistic situation. Let's say. For me, it's easiest to talk about Guanabens because that's ours, and it makes me feel on safe ground.
Yeah.
Let's say it works, and we have the registration and the marketing finished. I hope that we can give it to more patients than just to the ones who we did in the trial. I think that's a matter of debate because insurance companies will also have to say something about it, I think. But then there will not be like the strict inclusion criteria as in as for the trial.
Sure. And you have a healthy child, but they're just not walking type of thing. Is there going to be standards like in terms of that?
I don't.
Cognitively, they seem fine. They're doing well
Yeah,
at school.
yeah.
They have vocabulary.
Yeah.
But just not ambulatory anymore. Things.
I think
standards like that, I guess, is what I'm asking.
Yes, and if you ask me, that's not what I know for sure that we will achieve. But I would say if a child is in a wheelchair but is cognitive doing well, that's a perfect child to be treated. Mm-hmm. Because I think that walking is really not the most important function in life. It's communication and cognition. And so I would do that. And I would also change something else, like I cannot include pre-symptomatic patients in the trial. Mm-hmm. because you never know when the disease is going to start. And what I find very painful, and we had 2 brothers, one was pre-symptomatic and the other one was symptomatic. The symptomatic one could go into the Guanabens trial and the other one could not because he was not symptomatic, and we had just to wait until he became symptomatic, and then you've already lost something.
Right.
And if the drug is registered and you would make a diagnosis at birth, I would prefer starting treatment at birth.
Right. Yeah.
Because you may prevent any damage.
Yep. Yep.
And what is the rationale for not allowing pre-symptomatic treatment?
Because you then don't know the phenotype. So it's an open label study. It's not with placebo groups. So you don't know what to match with. We've seen very different phenotypes in one family. So they need to be symptomatic. We highly depend on the age of onset because that's such a good predictor that we depend on it. I see many different questions. Nicole.
May I add something to
Yeah,
the question?
yeah.
Because perhaps it was not what you meant, but in any trial you are allowed to stop participation.
Oh,
as a
yes.
participant If you find it too difficult or too long or too many side effects. So that's not a question. That's always allowed.
Yeah. And then we had the last question there because I think that there will probably be many more questions, but I'm really around. So I'm around for also to sit with anybody who would like to sit with me.
I might have missed that, but you're saying that you can't do prophylactic use. Is it because the phenotype determines what therapeutic you should give? Or is it because the drug has side effects?
No, we don't give it to pre-symptomatic patients. Because we don't know then when the age of onset will be.
But if the drug has been shown, I work in drug development, so I'm just trying to understand this.
But this
From the
is a
perspective of
trial.
Somebody who does safety testing on drugs, so if the drug has been shown to have been safe, I'm failing to see how you need to have a bad outcome in order to start a trial.
A bad outcome?
Yeah, well, you're saying that they have to have
Some signs of the disease.
Yeah.
So we go for patients who are still ambulant, but have symptoms. Because if you don't have symptoms, also it could be that the onset is at 12 years, which is,
Is there a problem with giving the drug for those 12 years?
Yeah, but outside the trial.
There's no way to measure. This is only for the trial. Once the trial is done, then that won't,
Yes.
then anybody can get it.
Yeah.
Only because you can't measure the trial.
Yeah.
There's no way to tell if it's working.
No, because it's an open label trial. I think I hate placebo treatment because if you, let's say, the treatment is for 1 year, or if you do 6 months and then open label, then still in 6 months, some children with vanishing white matter are devastated, and you don't get it back. So we felt like a placebo control trial is unethical. We go for an open label trial, but then we have to rely on historical controls, which we do have, but they depend on the age of onset. So I have no information at all. You're looking at me like you're not understanding it.
I was thinking, because I do drug development for cancer right now. And so I'm like, man, if there was a treatment that'd be helpful. then
But we don't know whether it's helpful.
the disease progress.
Yeah, okay. We don't know it's helpful. I think that we go to the next speaker, and if you wish to speak with me, that's fine.
And that's a good answer, is that you don't know. You don't have a
Well, we have in the Netherlands a committee that decides on such trials, the CCMO, and the CCMO told me that we have to assume that there's no effect at the beginning of the trial, and that you have to prove that there is an effect. And that's my job. Our job is to hopefully find an effect. If there is an effect, that we find it. And that's why you have to do the trial setup so strict, because of the chance that you otherwise don't find the effect, prove the effect, whereas it might even be there.
it's possible if the trial goes, it shows it's efficacious and it's safe, you could start people with open
Yeah, definitely. Then you can start them. But I was just saying, I would like to start them from birth. So I think then you have a perfect case for neonatal screening and start to treat patients from birth on.
Thank you so much for the invitation to speak, and apologies for the technical disruption. So it's a pleasure to be able to talk to you from the University of Wollongong in Australia. And I'm going to talk about some of the research that we do in my lab, which is focused on disease modeling and drug discovery. And in particular, the way that we, the way that we approach drug screening is by using patient cells. So we are trying to learn what we can from patient cells and do drug testing specifically in patient cells. So I'll talk about what we can learn from patient cells and the findings that we have so far. So when we're thinking about trying to understand a disease in a dish, we're obviously trying to represent the tissue by expressing different cell types. And as you've heard from the lovely presentation so far, in the vanishing white matter disease patient cells, we're trying to understand a combination of different cell types. But in particular, what you've heard is that there appears to be this big impact on one of the cell types in the brain called the astrocytes that have this really important role in supporting neurons and oligodendrocytes, but they also have other roles as well. So in particular, the purpose for doing drug testing in cells is that it can give us an approach to be able to test thousands of compounds. And in fact, this is what we've been able to do in patient cells. And then once you have those top hits or those top compounds, they need to be followed up in animal models or in humans, so in vivo in the body. But the cells provide us with what is described as a reductionist system. So we're all composed of trillions of cells, and each of those cells expresses millions of proteins. And the data that I'm going to show you in a moment focuses on a type of technique called proteomics. And that analyzes every single protein that is expressed in a cell. And as you've heard in vanishing white matter, there are big differences in how vanishing white matter astrocytes express different proteins as a response to this change in translation in the integrated stress response. So we're trying to understand how the whole proteome, the entire complement of proteins, is different, and then when there are drugs that we can identify, how those drugs then reverse that effect in those cells. So in the system that we use, what happens is a patient would donate some cells, and that can be something like skin cells. So a little biopsy is taken of the skin. Those skin cells are then plated into a culture dish, and they're given some food, some basically carbohydrates, proteins, and lipids, and those cells can then exist in that tissue culture dish as fibroblasts. And those cells we can then convert into what are called induced pluripotent stem cells. And that gives us the opportunity to then convert those cells into cell types of the brain. So in this study, we want to look at astrocytes. So we generate astrocytes in the dish, and then we can use those to do our in vitro, so our in the dish experiments where we can test drugs on cells. So it gives us the opportunity to make astrocytes from patients and culture them in a dish to be able to do our drug screening. So just a little side note on what has been done in other diseases using stem cell studies. And there are a lot of different diseases that for which this kind of approach has been taken. But I've just put together a little snapshot of some of the clinical trials for motor neuron disease through drug discovery using patient cells. So there are a number of different studies that have identified new drugs for disease using this kind of disease modeling approach. And what those studies tell us is that the screen design and the drug mechanisms are different for each drug that have been identified. But how they've been identified is to use different types of stressors to identify drugs that can prevent and protect the cells. And you heard in the previous talk about the importance of the role of stressors in causing kind of the phenotype of the disease. So what about in the patient cells? And again, we've already heard about the integrated stress response and the critical role that the EIF2B complex, those EIF2B proteins, play in controlling that integrated stress response. So all of those different types of stressors that you can see at the top of that pathway, they can all give rise to the integrated stress response, and that is what is impacted in the cells that come from the VWM patients. So when we look at cells, these astrocytes that come from patients, specifically in a dish in response to these stresses, what this data shows you is that those plots in red, which are the astrocytes from the vanishing white matter patients, are shifted to the left. So the data shows that with an increasing concentration of the stressors, we get a reduced viability of the astrocyte cells. And there is more of an impact. So there is a bigger reduction in viability of the astrocytes in vanishing white matter cells compared to people that don't have the disease. And this data also tells us that there are different types of stressors that show that effect. So the top one is hydrogen peroxide, the middle one is Mg132, and the bottom one is thapsogardin. And each of those show impacts in both oxidative stress and also something called endoplasmic reticulum stress. And the endoplasmic reticulum is a part of the cell that's really important for modulating protein expression. So the other plots on the right show us that when we stress the cells over time, those vanishing white matter astrocytes show alterations in the expression of those key proteins involved in the integrated stress response. So GAD34 and CHOP, which you heard about already. So it tells us that the VWM astrocytes are more susceptible to cell stress, including oxidative, proteasomal, and ER, endoplasmic reticulum stress. So again, you've already heard this morning about the astrocyte markers that appear to be different in vanishing white matter cells, and you saw data from mouse models and from humans, and this data shows you that the same is true for the astrocytes that come from patients that we've cultured in a dish. So when we're looking at different cell types, we use this technique called immunocytochemistry to specifically look at the level of expression of these specific markers. So there's an earlier cell type than astrocytes, so earlier in development, called neural progenitor cells. And these are the cells that then will go on to give rise to astrocytes in the brain. And those neural progenitor cells, they have expression of proteins like nestin and SOX2, whereas the astrocytes should show expression of GFAP, which is glial fibrillary acidic protein. And there's some other markers you can see there. So when we culture the cells in a dish, we should see expression of those markers specific to astrocytes. And what these plots show you is that in the cells that come from the VWM patients, they show reductions in the expression of those astrocyte markers. So a reduction in expression of GFAP and this other marker EAAT1. And that again is consistent with what has been shown in both the mouse models and in the patient tissue as well. So the other thing that we're able to look at is the response to stress, those stresses that we heard about already. So one of the stresses in that integrated stress response is the response to viral infection. And in the astrocytes that are cultured in a dish, we can give them a compound that mimics viral infection, and that is called poly-IC. So normally what happens in astrocytes is that when you give them this compound poly-IC, it stimulates a response to that infection, which a key marker of that is called interleukin 6. So in control cells, what normally happens is that we get an increase in interleukin 6 as a response to poly-IC. But that response is diminished in the vanishing white matter astrocytes. So again, this tells us that those astrocytes show a diminished response to infection. So overall, we see a loss of development of those astrocytes and a loss of their functional responses. So in this initial drug screen that we had done, we looked at several thousand compounds to see their impact in protecting those astrocytes against those stresses. And two of the compounds that you've heard about already are guanabens and ISRIB. And importantly, they did also show astrocyte protection in this screen. So what we're trying to do is identify drugs that can protect the astrocytes and promote their maturity so that they can reduce the inhibited immaturity and demyelination of the oligodendrocytes. And we identified a number of compounds that appear to be able to do that in the astrocytes. So guanabens and ISRIB importantly show that in this screen, but there were other drugs as well, and they may be possible to be used in combination with other drugs in the future following further testing. So we saw within that screen a lot of glucocorticosteroids, but they're not really a good plan for long-term use. The biggest impact that we saw was through a compound called edaravone, and that has been shown, it's already in use for motor neuron disease in certain countries, including the US and Japan. And it's thought that it's able to reduce endoplasmic reticulum stress and reduce reactive oxygen species. So it acts as a scavenger. So it mops up reactive oxygen species in certain cell types. And we also saw other compounds that are called ion chelators and bile acids. And they've been mentioned so far as well. So just a little bit on the way that we're approaching this. So we wanted to look at the whole proteome, as I mentioned, so trying to understand all of the proteins that are affected in cells. And you can think about the proteome as a network, because lots of those proteins within a cell will work together to work on specific pathways, specific functions that they need to perform in the cell. And by looking at all of the proteins together and the pathways that they target, we can start to see the kinds of pathways that are affected in the disease cells and that then are reversed in response to certain drugs. And so I've shown on the right this particular structure within this, so it's a representation of what a cell looks like. And there's this structure called, and it sits around the nucleus, it's called the endoplasmic reticulum. And there's also within the cell these structures called mitochondria that are responsible for generating energy. But as a byproduct of generating energy, they also create reactive oxygen species, which in themselves cause stress. And those different parts of the cell are going to be important when I talk about the data that we have. So what the data looks like when we do these proteomics analyses is that we're able to see changes in all of the proteins in cells that come from patients compared to controls. And we can put those into a specific piece of software that allows us to see how all of those proteins interact with one another, kind of like a network. And this is what it looks like. So that image on the left-hand side is every single protein that is different in the patient cells. and how those proteins are connected with one another. And we know how they're connected based on the pathways in the cell that they're affecting. So the major pathways that are affected in the vanishing white matter astrocytes are shown on the right-hand side. So the bigger those bars, it tells us the more proteins are involved in that pathway and are affected in vanishing white matter disease. And what is shown in the pathway is, in the map, is this cluster of proteins that are affected in vanishing white matter that seem to be associated with mitochondrial dysfunction and oxidative stress. And the other thing it showed is that there are big differences in the energy production pathways within those astrocytes. So one of the energy production pathways that's really important in astrocytes is glycolysis, and that appears to be affected. And the other major way that, that astrocytes generate energy is through a process called oxidative phosphorylation, and that also is affected, and that is part of the mitochondrial pathway. So those arrows show you a few of the mitochondrial pathways that appear to be affected in the patient cells. So overall, what we can see is that the whole proteome is affected in VWM astrocytes. Lots and lots of pathways are affected. And this is called a volcano plot, and it shows all of the biggest changes in proteins, and they may either be upregulated or downregulated. So we may see an increase in expression or a decrease in expression of those proteins. Overall, it tells us that there are lots of proteins affected, and we can then use this data to look at the major pathways. So some of the pathways that I'm going to pull out for you now are around those specific functions. So as I mentioned, the mitochondria, they generate energy, which is a really important function, but they also generate reactive oxygen species. And we can see that the vanishing white matter astrocytes show this downregulation of antioxidant and metabolic proteins, so proteins that are important during the process of metabolism, which is what's necessary to make energy. We can also see there are big changes in not just the mitochondrial proteins, but also the ribosomal subunits in the astrocytes as well. And the ribosomes are also important for protein translation, so making the proteins. So if we're going to try and reverse those processes that we've looked at, there's a couple of different strategies. So whenever we're trying to address preventing some kind of pathway from happening, it's usually a good approach if we can try to do that using two completely distinct approaches. So we did that by following up on that data with edaravone, which, as I mentioned, is able to prevent endoplasmic reticulum stress, and it's also a reactive oxygen species scavenger. And we also used a completely separate approach, which was to take healthy mitochondria that we can isolate from healthy cells and directly transplant those or transfer those into cells, into the astrocytes. And this is an approach that's been done for other neurodegenerative diseases that show deficits or show impacts in mitochondrial function. And it is something that is being explored for direct transfer into people, and that is being tested at the moment for other diseases. So to look at what the impact of these approaches on the proteome is exactly, again, we can go back to our pathway analyses, and all of those red dots that you can see in the pathway are after the treatments with these approaches, the increase or the protection against that pathway of oxidative phosphorylation. So lots of those proteins that are affected in oxidative phosphorylation are then prevented with these treatments. So we can look specifically at all of the proteins that are affected in the treatment. So when we treat with edaravone, there's 146 proteins that are modulated by that treatment. Importantly, they appear to show benefits in EIF2 signaling, which is that central component of the integrated stress response. They also show reductions in what's called the unfolded protein response, which is important in ER stress, so endoplasmic reticulum stress. And the cells also showed improvements in oxidative phosphorylation. The effect of edaravone appears to be quite specific in actual fact in terms of protecting these metallothionein proteins as a way to protect against oxidative stress specifically. And the plot on the right shows that edaravone can reduce the presence of reactive oxygen species. Now, when it comes to the mitochondrial transfer, we can monitor that using using a fluorescent reporter. So in the plot on the right, the yellow color shows us when the mitochondria.
I would like to thank the organizers of the meeting for allowing me to share some research ongoing in my lab about development of a gene therapy for vanishing white matter. I've been a previous participant of ULF, but in the space of Krabbe disease, where I've worked for the past 9 years developing a gene therapy for Krabbe. Over the past 2 years, we've gotten 2 gene therapies successfully into the clinic, and it's allowed me some space and time to really take this passion and move it on to other very deserving diseases like vanishing white matter that have a lot of commonalities in these leukodystrophies. I do want to point out that this is a collaborative project with Joshua Bonkowski at the University of Utah, and he's joining us on Zoom. I don't know if he's with us yet, but it's a real privilege to be in the room with many of you and meet you for the first time in person. So we've already had a really great background on the genetic basis of vanishing white matter, so I really won't belabor this, but just to remind you that vanishing white matter arises from mutations in 5 different subunits of the EIF2B complex. And today I'm going to focus on EIF2B5 because that is the subunit where the vast majority of mutations are found. So this is kind of a roadmap that I follow in my therapy development in my lab. I strongly believe that you really need to understand the mechanism of disease before you can develop a therapy that's really appropriately targeted to the right pathways and cell types and brain regions. What else is necessary is that we have the correct model systems, because we cannot go directly from cell culture dishes to a human patient. It's really important to me to characterize these models and find appropriate clinically relevant readouts so that when we go to translate a therapy to the clinic, we know that we are hitting those important readouts. And so then the fun part for me is the therapy development and then really trying to determine efficacy and dosing and the appropriate animal models. So we have heard a lot today about astrocytes. I don't think I need to prove to you that astrocytes really are central in the pathomechanism of vanishing white matter. We've had some really important groundbreaking work from Dr. Vandernap here that have proven this. We also heard a really great talk yesterday from Dr. Elroy Stein showing that the energy homeostasis in astrocytes is really the Achilles heel of vanishing white matter disease. So we know that astrocytes are central in the pathomechanism, which also makes them the key therapeutic target. So we are working on some in vitro modeling. It's a little bit similar to what the last speaker talked about. In my lab, we can get patient fibroblasts, which is from a skin biopsy, and use direct reprogramming to make astrocytes. And this project is led by a very talented PhD candidate in my lab, Jessica, who's here with us this week. And we have made VWM patient-derived astrocytes in our lab now, thanks to generous donations from patients, and we are trying to study more about the disease mechanisms of these very critical cells in vanishing white matter. So with the understanding of disease mechanism and the astrocytes are central to the pathomechanism, the next highlight in my lab has really been working on this model characterization. The very well-known classic R191H model, Dr. Vandernap already introduced this very eloquently, so I won't go back into this, but just to know that this is one mouse model that really recapitulates a lot of the disease, myelin loss, ataxia, the motor deficit. She showed that very nice catwalk data and has a shortened lifespan. There has been a newer mouse model.
It was working. Okay, this is a newer mouse model that was first reported in 2020, and it's called the I98M toy mouse. And this results from a mutation in exon 2 of EIF2B5, and just so you know, why are they called I98M? It's the isoleucine to methionine change in the protein at position 98. So that's why we call them that, just for a little information about why we have these strange names for our mice. And this mutation occurs within the catalytic subunit of EIF2B5. And this is a really pretty severe mouse model. You just saw how tiny that mouse was. That was at only 4 weeks of age. So when we track their weight over time, we see a really dramatic reduction in weight in both the males and females compared to the wild type healthy male and female mice. We also see a very significantly shortened lifespan of about 150 days in these mice. So they live for only about 5 months of age. They also have seizures, and they have that ataxic gait. Currently, this is the most severe vanishing white matter model that's available. However, there are probably other models that are coming in the near future. So this is an example of a test that we do called rotarod. So here on the left, we have a wild type mouse at 4 weeks of age, and then an affected vanishing white matter mouse at just 4 weeks of age. So this rod starts to spin, and we try to see how long they can hold on. So you'll see here the wild type mouse can keep going, and within 20 seconds, the affected mouse falls off the rotarod at just 4 weeks of age. When we look at this out to 150 days, we see an even more significant reduction in the rotarod with the latency to fall of less than probably 5 seconds. So again, this is one of our readouts that we can use showing how severe this new I98M mouse model is. We already had a nice introduction to the catwalk by Dr. Vandernap, so we have the same equipment at my institution. And here again, we see that really nice mouse walk, and this is the footprint map that it reads out. It also reads out the stride length, the footpaw intensity. There's so much information that we can get from this catwalk. And then when we have our affected I98M mouse, this one's about 4.5 months of age, very similar to what you saw earlier with the R191H. It leans against the side. It has a wide base stance. It's ataxic. It's shaky. And when you look at this map down here, you see a very clear, really dysregulated gait compared to what we saw with the wild type mouse. So we've been working to characterize this gait and the rotarod, and we also have a high-resolution 9.4 Tesla MRI for mice. We know that MRI is really critical in the human patients in diagnosing and then tracking their disease progression over time. So we're doing the same thing in the mice. So in a wild-type mouse here, we see a nice myelination pattern, and we see very significant reduction in the myelination, particularly in the corpus callosum, but other brain regions. We're also doing DTI, which is diffusion tensor imaging, which allows us to actually quantify how the water molecules are moving in the brain and get some more quantitative measures from that. And we're also seeing differences on DTI. So again, a clinically relevant way to look at this mouse model so that when we can pick up changes with a therapy. So then we also look at the brain tissue. We know that there's loss of myelin in this disease. So this green stain is a stain called fluoromyelin, which I paired here with a neurotracer. And we see in wild type mice, you have this really nice green stain where you should have myelin in the brain. That's also present in the het mice, which are the carriers. However, when you get to our affected mice, we see almost complete loss of this fluoromyelin in the brain. So this is very similar to what we're seeing on the MRI, that there's severe reduction in myelin on MRI and postmortem histological analysis. So now I've hopefully shown you that we know that astrocytes are the real pathomechanism of disease and the targets. We have this new model that was just identified in 2020 that we've been working hard to characterize using clinically relevant readouts. And now the really fun part for me is getting to a therapy development. So our therapeutic premise here is that we know that there's genetic mutations in EIF2B5, and this results in loss of function of this critical protein. Then you have stress-inducing events, fever, infection, or trauma that results in ISR activation and cell death and loss of myelin. So our therapeutic premise here is called gene replacement therapy. So we have a virus known as adeno-associated virus that's a delivery tool to get a healthy copy of the EIF2B5 gene. So what AAV does is it gets into the cell. It exists as an episome, so this little circular piece of genetic material. It does not integrate into your DNA. So we're not trying to fix the mutated copy. We're simply providing a healthy wild-type copy that will make a functional mRNA and then make a functional protein. So this is a gene replacement therapy. It's not gene editing. We're not targeting the mutant form that's already present. So we're hoping that we can compensate by providing a healthy copy of the gene, which will then turn into a healthy copy of the protein. So just very briefly, what is AAV? When you have a wild-type AAV, that just means like the virus that's actually occurring. There's only 2 genes in it, rep for replication and cap for capsid, so it's very simple. And when we're using it as a vector, we take out both of these and we put in our gene of interest, which of course is EIF2B5 in this case, and then a promoter, which is what drives where the expression of that gene goes. And then we have other regulatory elements here as well. So for us, it's really just putting this AAV with this EIF2B5 and trying to regulate it with these other components. AAV is known to be non-pathogenic, so we've all been exposed to it and it doesn't make us sick. It rarely integrates, so that's a good safety profile because it doesn't get into your DNA, which could cause off-target things like cancer. There's low immunogenicity, has pretty broad tropism, and it's targetable. We can use these different components to drive it to where we want it to go. It's pretty small, which is good and bad. It's good because it can cross the blood-brain barrier, and there's a relative ease of manufacturing. It's been in over 250 clinical studies now, which has a pretty good safety profile, and it's FDA approved for the treatment of spinal muscular atrophy, which has been dosed in over 2,000 patients now. There are some limitations, of course, and one of the biggest ones is that it has what we call a packaging capacity of only 4.8 kb. So 4,800 base pairs of genetic material can fit inside this entire construct. So if you have a gene that's really big, then it might not fit inside of this particular virus. So AAV comes in a lot of different flavors, if you will, or serotypes. Guangping Gao is here at this meeting, and he is actually the pioneer of AAV and discovered the vast majority of AAV serotypes. With these different capsids, they have the tissue tropism or a propensity to go into different tissues. So depending on what tissue you want to target, you want to choose a different variety of this adeno-associated virus. So we're obviously interested in the brain and the spinal cord. So for our study, we selected AAV9 serotype, which was also the one used in SMA. So since the FDA has already approved that, it has a more direct path to the clinic. So with now knowing which capsid we want to put on the outside, what do we want to put on the inside? Obviously, our gene of interest is EIF2B5, but then what about this promoter, this driver? It's been shown that you can swap out this promoter and drive your expression of your gene into different cells. For example, you can put in a promoter known as CBA or chicken beta actin, and it'll go primarily into neurons. You can put in myelin basic protein, and it'll go primarily into oligodendrocytes. Or you can put in this GFAP that we've heard a lot about, this glial fibrillary acidic protein, and it'll go primarily into astrocytes. And it's been shown that you can drive this expression almost entirely, almost 100% into astrocytes at 2 different time points by sticking in this GFAP promoter. So now you know what capsid we want on the outside. We chose AAV9. You know what promoter we want on the inside. We want to get into astrocytes, so we're looking at this GFAP promoter. And then now, where do you want this virus to go? So it depends on really what organ systems you're trying to target. So we can give gene therapy systemically or by intravenous injection, and it will go throughout the body. Some of it will get into the brain if you use blood-brain penetrant AAV serotype like AAV9, but not as much of it gets into the brain if you give it in the blood. And also you have to think all that virus gets into the other organs in the periphery, namely the liver, and we've seen toxicity in the liver, especially with really high viral payloads. So what we focus on is the cerebrospinal fluid, where we give it directly like a CSF tap. Then it bathes the brain and the spinal cord, but really limits the off-target effects of the liver and other peripheral organs. We can also use organ-targeted gene therapy. You can target specific regions of the brain. You can give it directly to the eye, the lung, joints, or muscles. And this is usually used in areas that it's difficult to get the virus into by giving it CSF or intravenously. So what did we want to do to try to develop this gene therapy for vanishing white matter? We looked at 3 different promoters, that ubiquitous one that I mentioned, and then also GFAP, which we've seen from previous literature that it really goes into astrocytes, so we were most excited about that. However, GFAP is really big. It's 2,200 base pairs. And remember, I told you that AAV has a packaging capacity of only 4.8. So now you're taking up about half of the virus with just your promoter, and you have all that other stuff to fit in, namely the EIF2B5 gene, which is also over 2,000 kb. So we were really worried about this really exceeding the packaging capacity of AAV. So we looked at a truncated GFAP called GFABC1D, which takes out a number of the functional domains of GFAP, but has shown to still be expressing. So we injected these packaged them into AAV9, injected them into little neonatal mice. We grew them up for 4 weeks or 8 weeks, and then we looked at the brains to see, okay, where did this virus go? And because this is a green fluorescent protein that we put behind these promoters, we can literally see glowing cells in the brain, which is really cool. This just allows us to figure out if we're getting the virus to the right brain regions and the right cell types. So what did we see when we took down these mice and looked at their brains? We saw really beautiful transduction with this GFAP all the way. We see here in the cerebellum, the back region of the brain that we know has a lot of those mislocalized astrocytes that we talked about, all through the white matter here, even to the most forward regions of the brain. So we were really excited. This looked really pretty to us. The truncated version, that's 681 base pairs, we lost quite a bit of expression, but it still looks nice. I think I would have been really excited about this if I hadn't seen what was like really possible. But one thing we were really concerned about is that it really poorly targeted the cerebellum that we know needs to be a target for vanishing white matter. And then our ubiquitous promoter really didn't have great expression throughout the neuroaxis. And when we quantified this expression, not surprising because it's just what our eyes see, the GFAP, the full-length one, had the greatest expression throughout the brain. And then we can even look at these brains glowing, which is pretty cool. So this is in a different order, sorry, but this GFAP here is the full one. You can see how bright green that brain looks compared to the truncated GFABC1D. Then we can actually take the brain and measure the amount of protein that's in that piece of the brain by doing what's called a Western blot, where we use antibodies. So for our GFP here, you can see that there's the most protein. This is these kind of blobs here on the membrane for GFAP compared to GFABC1D and our ubiquitous promoter. So this is just using multiple methods to really prove the same point about which promoter is really doing the best at driving expression. And so we also, of course, need to make sure that this is getting into astrocytes, right? We saw the whole brain glowing, but then we need to zoom in and look and say, okay, are you really getting into the cell type that we need? And we really see beautiful transduction of astrocytes with our 2 astrocyte targeting promoters. And with our ubiquitous promoter, not surprisingly, we're hitting both neurons and astrocytes, so it's less specific. We have started to quantify this. We only have 3 mice quantified, but we're hitting less than 5% of neurons with our astrocyte promoters, so it's really not very leaky. And almost all the cells that are green are astrocytes, are cells of interest. So what we've done here is really trying to prove our therapeutic premise that that we have an astrocyte targeted gene therapy. This is just a pretty cool video that we can take of an astrocyte really zoomed in, showing that green inside the astrocyte and the processes, so showing that we were able to get our virus and our protein into our cells of interest. So now we've understood the disease mechanisms, we've characterized the model, we think we have a therapy to develop, and now it's time to put these into our models that we've characterized. And so we took the same constructs, we took out that fluorescing protein and put in EIF2B5, our transgene of interest. We packaged it into AAV9, and first we tested this in the R191H model. So this was done at University of Utah with Josh Bonkowski. He has this model, so I sent him our viruses, and he injected them. And we grew up the mice, and then we looked at them by rotarod and gait analysis. Then we looked at their brains for morphology and histology, quantitative PCR, and Western. This study's still ongoing, so I'm just going to show you some of our earlier data today. So what happened when we look at these mice on rotarod? Well, we were really excited about this full-length GFAP construct that looks so pretty in the brain, and it had very negligible impact on the mice on rotarod. You might think, why is that? Well, this is the exact concern that I had, that when we put all this into the virus, the GFAP and the EIF2B5, the total amount of genetic material we put in was 4,500 base pairs, which was exceeding the packaging capacity. So we got a very bad production of virus. The yield was poor, the titer was poor. There was a lot of capsids that were empty. They didn't contain our gene of interest because we tried to shove too much in there. However, if you look at our truncated GFABC1D, and this is out to 6 months in these mice, we see normalization of the rotarod. So they're really significantly improved compared to the sick mice that didn't get treated, and they're not significantly different from the wild type mice, the healthy controls. And we're up to over 20 mice in each cohort now, so we have decent animal numbers for this study. So then we took the same 3 constructs and we injected them into our severe I98M mouse that I showed you earlier, and we did the same analyses that they were doing in Utah. We were doing this in my lab in Columbus, Ohio. And we are further behind in our study because we injected the mice at Utah first while we were characterizing our model. So we have pretty small animal numbers here, only 5, 8, and 6. But so far, the numbers are trending similar to what we see in Utah. So we do have a significant reduction with this truncated one. Again, the full-length one didn't work because it was a bad virus. We put too much genetic material into it. And the ubiquitous is having some improvement, but not as much as the small astrocyte one. So we're seeing comparable results between 2 mouse models, which I think is going to strengthen our study design overall. However, I think that I was really kind of hung up on how pretty this GFAP looked, and that this GFABC1D, the truncated, isn't really fully correcting the disease. When you look at survival, this is actually pretty exciting. Our red line here is our affected. We did a sham treatment group, and those are the gray bar here. There's obviously no difference. Sham is just when we give the vector diluent, and there's no actual therapeutic efficacy there. The pink now is the full-length GFAP. They're dying pretty quick, not surprising based on how they did on the rotarod. And then the CAG, the ubiquitous, those are, every drop in the line is a death. So we're seeing some extension in survival. However, that truncated one that was looking good on the rotarod, we've only had 1 mouse die, this 1 drop, and the rest are still ongoing beyond 200 days. So we're pretty excited that we've pretty much doubled the lifespan of these mice with this therapy. However, I really want to try to make the best construct possible, and I think that with that 681 base pairs, we maybe took out too much of what was in the full-length GFAP, and I was like, we can really custom tailor fit a promoter that will go just with EIF2B5. So we put back in more of these binding regions. We put back in C3, C5, C6, and we kept out all the inhibitory regions to try to make this novel astrocyte promoter. And when we took down the mice and we looked at these brains, they look beautiful. So we have a lot of transduction throughout the brain, and we actually got this expression back into the cerebellum, which is what I was really worried about with that truncated GFA, the one that was only 681 base pairs. This very nicely targets astrocytes, and you can see here just it's really throughout the brain, really robust signal. Now, when we look at those glowing brains again, compared to the GFAP, the full length, and the truncated, we see how bright green these whole brains look. So we really did restore the level of expression by putting in those 3 extra regions. And when we compared the amount of green, the amount of expression in the brain between all 4 promoters, our new promoters actually, we have 10 mice taken down now, is actually significantly higher than the full-length GFAP. And you might say, how did you do that? And once again, we took out inhibitory regions and put back in those 3 regions that were missing from the truncated version. So next steps is that we want to evaluate this novel astrocyte promoter in both mouse models. I have to leave you a little bit hanging here because we just made this new promoter and just took down our green mice, and we've injected both the Utah mice and the mice in my lab, but they're really young. So we don't have data yet, so hopefully next year we'll have some data on how this new promoter is looking in both mice. And really the goal is to come up with a lead construct. You know, is this new promoter going to do even better than this? You know, will our mice live longer, and how well will this promoter do? We don't know, are these mice going to die next week, or will they live for a year? We're really not sure at this point, but we're pretty excited about this, and we think with this increased expression that we might see even greater effect with our novel promoter. After we determine our lead construct, we'll do a dose escalation study, and then we've also been working with Paul Taysar at Case Western. We have patient-derived organoids now, and we'll test our lead construct in these and the patient-derived organoids to make sure our expression is really there in the context of human cells, which is important in the translation to the clinic. So I hope today that I've shown you that we have a really good understanding of the disease mechanism thanks to all the work that's gone into this for really decades. And we have characterized now multiple models of vanishing white matter, which is a big advantage over other diseases where there aren't good models of disease. We've developed therapies, and we've started the efficacy testing of these therapies in 2 models. And then we still need to perform our dosing study to figure out what our lead construct and our minimal efficacious dose is. You might say, what's next after that? Then we do safety and tolerability studies and hopeful translation to the clinic. We've heard a lot about combination therapies, and I think with a disease as complex as vanishing white matter, it might be a combination, approach. It could use one of the other compounds to help stabilize disease before giving a gene therapy, which is a one-time treatment. You're delivering a healthy copy of a gene, so it's a one-time treatment. So this is really kind of what I think of as my patient-centered preclinical study design. It's really all about the patients using relevant in vitro and in vivo models, developing a therapy that's really targeting those specific disease mechanisms, making sure that we have, first do no harm, that we do our safety studies and have regular interactions with the FDA. And then have concurrent natural history studies and registries, which is beautiful work that's been done by the Vanishing White Matter Consortium, really setting up for the success of clinical trials. So this is my lab group. We moved into a brand new lab building this week, so that's been fun but challenging. And again, Jessica's here. She's the PhD candidate that's been really running all of these studies. Couldn't have done it without her. And then the collaboration with Josh at the University of Utah. So thanks again for your attention and having me, and I'd love to take questions.
Hi, thank you very much for the presentation, and I think it's super exciting results. My question is actually because I'm so excited. So you're now focusing on EIF to B5, and of course because you have a model, animal model for EIF to B5. If this proves successful, would you consider also doing EIF to B1 to 4?
Absolutely, yeah. I think there's a model of EIF2B4. The next most common mutations are in EIF2B2, so I think it's a natural next step is to take out the EIF2B5. We already have the vectors, and yeah, try it in different animal models with different subunits and hope that we can do the same. I mean, each subunit works a little bit differently, so I'm not going to say that if it works for EIF2B5 that it would work for the other ones, but hopefully, that would be definite next steps.
And then would you need to generate mouse models for all these different subunits, or?
Some of them already exist, so I think that would be kind of the easiest next steps. But with gene editing now, we can use CRISPR to make mouse models, and the timeline's really only about 9 months of a patient-specific mutation. So we can really start to make models, generate them, and characterize them in less than a year, which is light years ahead of where we were just 5-10 years ago. And we no longer rely on knockout models, which we know for proteins that are this important, knocking them out, they the animals aren't viable. So yeah.
Thank you.
Yeah.
Very nice talk. Have you looked at the modulation of the ISR signature with these constructs? Because that could be a really nice way to know how much you need to administer. Maybe already there.
Yes, so this is a very good point. So in both of our mouse models, we've now done extensive PCR on the qPCR on the untreated model. So we have a whole panel of the ISR. We're looking at ATF3, 4, 5, the TRIB3, and the CHOP. And in our untreated mice, we see significant elevation of these markers. So now as we're starting to take down our treated mice, we're running those PCR. It's just a little bit preliminary for me to show any of that data right now, but it's in the works.
Thank you so much, Alison, for your beautiful work, and it's really fast. Just to moderate expectations, because I was speaking to a parent yesterday, and I had the impression that the parent thought that gene therapy would completely cure the brain of a patient who is severely handicapped. And would you agree that the answer is the same, that you cannot get back, if there's a hole, there's a hole?
Exactly. So what we've seen with, I really think almost all of the gene therapy trials is the earlier the intervention, the better. Sometimes the FDA actually advises us to start with slightly older population to make sure that it's tolerable before they allow us to move to the younger populations. But both earlier intervention and usually higher doses to a certain extent have really had a better outcome for gene therapy. And what I try to remind patients is that the virus has to get into a healthy cell to work, right? So you have to have these cellular targets for the virus to get into. If you've lost a lot of those cells, or if the cells aren't functioning, getting virus into them isn't possible because there's not the cells to get the virus into to bring that delivery cargo and make the protein. And if the cells aren't healthy and they can't function, then they won't be able to make that protein. So the virus can only do with what targets it has in the brain. So I think you're exactly right. And we also don't expect reversal of disease. It would be more of a stabilization. The only kind of glimmer of hope that I have from the Canavan study is that they saw some spots of what they're calling some improved myelination after gene therapy. So perhaps if you can stabilize the brain, maybe it will give it a chance to improve myelination in some areas. But I think that's kind of the best expectation. So I think for all these diseases, getting in the treatment earlier is better. But exactly, what you said too, every family has different expectations and what they want. And for some families, stabilization and to be able to retain some communication, even if they never will walk, is a really meaningful outcome to that family.
Yeah, no, I totally agree with you. It's just that the expectation need to be set at before the therapy. That people don't have the expectation that in wheelchair-dependent person will walk again, and which is not going to happen.
And I will also be around, not for the whole night, but until about 7 o'clock. So if anyone wants to sit down and chat with me or have any more questions this evening, just come catch me in the next couple hours. And then you can also I'm happy to give anyone my email address. And if you're ever in Columbus and want to stop by and see our lab or meet anyone, you're more than welcome.
Thank you so much. Thank you.
Hi everyone, it's finally nice to meet you in real life, or finally it's nice to meet you here. So my name is Romy van Forst and I'm from Amsterdam UMC. I'm part of Dr. van Knaap's research team. And I'm actually here talking, presenting this project we're doing, which is the impact of vanishing white matter on non-affected family members. So this is more a talk about actually you as family. And this project is in collaboration with the Children's Hospital of Philadelphia, the McGill University, the University of Tübingen, and the Amsterdam Leukodystrophy Center. So, yeah, you probably already all heard it, but I give you a quick recap of what vanishing white matter is. So vanishing white matter causes chronic neurological decline, but also has these acute episodes of rapid decline, which can be triggered by certain stressors, such as head trauma. And I can give you a clinical definition of what neurological decline is, but I thought it would be better to give you an overview of some of the symptoms you mentioned to us during, for example, interviews, but also the registry. So here you can see the broad variety in symptoms. So difficulty in walking is a major one, but also, for example, anxiety or irritability. And these symptoms, or vanishing white matter, is actually caused by a genetic defect in the AF2B, as Mario already mentioned. And because it's a genetic rare disease, you would expect that the disease progression is somehow more predictable. But unfortunately, this is not the case. As you can see here in this graph, Mario already showed it as well. You can see on the y-axis the health index, and on the x-axis the duration in years. And if I give my pointer, if you see here with the onset of less than a year, there's a very steep decline and there's not much variability between patients. But if you look at the other onsets, you can already see that there is so much variability between patients that it's very hard to predict what the disease progression is. But it is in some degree related to the age of onset. And this variable in disease progression, but also the symptoms you have mentioned. Okay, all right, thank you. And the symptoms you've mentioned indicate that vanishing white matter patients often need different types of care throughout their lives. But what we haven't investigated yet is what this actually brings along on the family members. What is the impact of this on family members? So I want to say, I want to explain to you why that is important. Well, first, we want to raise more awareness that care should not only be there for patients, but also for family members. And this is something which is often overlooked by healthcare professionals. But in order to say care should be there for other patients, and we have to raise more awareness about the impact, we first have to measure what the impact is. And this has not been done for vanishing white matter yet. Further, it's measuring the impact can help us develop these impact outcome measures, which indicate treatment effect. So you can see before the treatment and after the treatment if there is different scores in family functioning, for example. And then, of course, it's a very good first step to develop a more tailored approach on the actual needs of vanishing white matter family members. So how do we want to do this? The study is still going, so if you feel inspired after this talk and you want to participate in the study, please come up to me or email me. I'm always open to any comments, feedback, or questions. But for now, we have included 94 participants, so parents, partners, and siblings, and we asked them about their quality of life, so the quantitative measures, which are more standardized questionnaires, and we have these qualitative measures in which we go over an impact of disease inventory, which are more open-ended questions about more specific aspects of the impact it has on family members. And of course, we have the nice registry in which we send out these yearly questionnaires for the disease progression of the patients, and then we have these severity scores, which we can correlate as well. So, as I mentioned, first, I want to say something about the quantitative measures. So here you can see the quality of life, or the Euro quality of life 5 dimensions, which measures mobility, self-care, usual activities, pain, discomfort, and anxiety and depression. And each domain is divided into 5 levels of perceived problems. So, for example, level 1 is indicating no problem with mobility, whereas level 5 is indicating extreme problems with mobility. And we also asked them about their health status, about how are you feeling today, in which participants can score their own health on a scale from 0 to 100, where 0 is the worst health they imagine, and 100 is the best health you can imagine. And for this study, we're now looking at the moderate to extreme problems. And these are the first results, and it's still a pilot study. And I also want to mention that, of course, every situation is unique, but this is something we found on group level. So here you can see the first results from all the family members, so fathers, mothers, partners, and siblings. And on the y-axis, you can see the VAS score, which is the question, how are you feeling today? And with 0, worst health, and 100, the best health. And you can here also see the standard population, which a normal person would score. And you can then see that actually all family members score lower than the standard population, and especially partners, which score significantly worse. I have to mention we only included 3 partners because financial environment is predominantly a childhood disease. This is very hard to include partners. But this is something we do see back in the clinics, where partners really have struggles with family functioning. So if we dive into the different domains, you can see that, for example, so the different domains to see the moderate to extreme problems with these domains, you can see that mothers really score lower on anxiety and depression, and pain and discomfort, whereas fathers and siblings score lower on anxiety and depression. And the partner score, this one has to be there, but the partner score lower on the usual activities, and these are the reference scores. So we also have another standardized questionnaire, which is the PET-SCORE-L, which measures different, also more in-depth domains, which is the physical functioning, the emotional functioning, the cognitive functioning, social functioning, communication, worry, daily activities, and family relationships. And if you add up these domains, you can get some summary scores, which is for the physical, emotional, and cognitive and social domain, the health-related quality of life. So it's more about your own health. And then you also have the family functioning score, which is more the daily activities and the family relationships. And here are, again, some of the first results. So here you can see the summary total scores. So you can see the fathers and the mothers and the standard population, which is again the dotted line. And 0 means again a lower score, and 100 means a better score on this particular domain or total score. And then you can see actually that on the total score, both fathers and mothers score lower. Fathers score a little bit higher than mothers, and this is the health-related quality of life and family functioning score, but they all score lower than the standard population. And if you look into, or you dive into these different domains, you can see that, for example, on the emotional domain, especially fathers score lower than mothers, and also they all both score lower than the standard population. And for daily functioning, mothers score lower than fathers. And for the social functioning, both parents score actually quite low. And then we also asked non-affected siblings these questions, but we only included the physical functioning, the emotional functioning, the social functioning, and the daily activities. And here are some of the first results. This was quite surprising to us, but you can see here the different domains again, and then the black line is the non-affected sibling, and the dotted line is the standard population. And you can actually see that they score a little bit higher than the standard population, except for the emotional domain. I have to say we only included 11 siblings, so this is not really representative yet, but it's something that surprised us. And yeah, we think that's also very difficult to find these true values, because if you want to talk to a stranger about all your problems, especially if you're in puberty, that's kind of hard. So we understand that, that they probably score a little bit higher. But yeah, we have to include more non-affected siblings as well to get more representative results. And then we had the qualitative interview, which has more open-ended questionnaires and we dove into the different teams. And there were some recurring themes per category. So for example, for parents, they often report their unique relationship with their child, but also social isolation due to, for example, the threat of an acute episode or the amount of caregiving they have to do. And change of career plans is also something they reported frequently, and the lack of expertise of vanishing web matter in a normal healthcare setting. And this is also something which is shared amongst partners. They mentioned it as well as an increase in caregiving because they have to take over certain tasks of their partner, which their partner normally does but now cannot do anymore. And then partners often also feel alone in their experiences because they feel like, because vanishing web matter is, of course, predominantly a childhood disease, there's no one they can really talk to and share their experience, thoughts, or emotions with. And then siblings mentioned their heightened social awareness, but also sometimes complex feelings like, can I be angry at my affected sibling? And sometimes a lack of attention because they feel like the other sibling takes up a lot of time for example, care. And there are some overarching themes which all family members share, which is the threat of an acute episode, which really impacts and changes their whole lifestyle. And also the anxiety for the future, because the disease progression is so variable, it's so unpredictable, so there's a lot of anxiety for that. But they also mentioned a more appreciation of life, that they feel like they have a more unique relationship with their family and appreciate more the little things in life. So this is just the first data, but it already shows that partners really have a low quality of life score compared to the standard population, and that they score particularly low in the usual activities, and that both parents score low on the social domain, and from the PET score L compared to the normal standard population. Mothers score lower on all domains, especially daily activity. Fathers and healthy siblings score lower on the emotional domain, and some overlapping themes from the qualitative interview are anxiety for the future, the daily threat of an acute episode, and appreciation of life. There's a spelling error in there, but I'm sorry for that. So you can already nicely see that these different family members have different scores on different domains, indicating their different needs. And bringing these different needs into light and raising more awareness of these different needs is already a very good first step towards developing a more tailored approach for that. So this is in short the project I'm working on. I'm still recruiting participants, so please, email me or contact me, because without you, I know I send out a lot of questionnaires and I ask a lot of questions, but without you, we wouldn't have been able to do this, as research is something we do together. So I also would like you to invite you to engage in a conversation with us. So if you have any comments, feedback, or questions, we're more open and welcome to. We're always open to them. And I also want to thank everyone who is involved, especially Dr. van der Knaap. So this is my project. Are there any questions?
Thank you for your talk. A bit of an academic question maybe. When you say standard population, do you refer to standard population in Europe, or is it standard population across the world? And how does that compare with your sample, which I'm guessing is a mixed demographic as well?
Yeah, so this is the first data. So we're now looking only at the standard population across the world. But if we do the real data analysis, we have to match, of course, on age, on sex, and on which country the participant is from. Yeah. But it was more like giving an indication on how the family members scored across, normal, what is considered normal population. Yeah.
Does it matter on your siblings? The data that you're collecting for the siblings, like my daughter's an adult, so her siblings are older.
Mm-hmm.
Does it matter because a lot of these may be for the younger children, or?
So we're including all siblings, no matter what age. But yeah, we have to look at different age categories as well, because if you're 16 or if you're 35, you probably have a different experience, or you reflect different on your emotions. So we have to look into that as well. But for now, because we have such a small or limited population, we just look at the overall effect. But we will dive into that. Yeah.
Is it not so that they have to live in the same household?
Yeah.
So they need to live in the same household, because if you live, if your other children, healthy children, are healthy and have their own new families, then the impact is of course very limited. So that's something, they have to live in the same household, right?
Yeah, but I also did some interviews with older participants, because they can reflect on the period when they were living in that household. So the questions are more reflecting backwards. Yeah. Yeah. So indeed, yeah, that's the inclusion. So if as the patient gets symptoms as an adult, so the sibling is not. Has not experienced it in this household, then we would not include this sibling. Yeah.
My name is Allison Bach. My son Sam is 12, and he was diagnosed with VWM when he was 2. And he is not here because I did not want to travel with him. but he's a very sweet little boy. So we started the VWM Families Foundation a few years ago, and in the last couple of years, now that clinical trials and things are coming up, we have decided, we also used to have the VWM Foundation that Brett ran, and Saving Chloe Saxby in Australia, and now that clinical trials are coming, we have decided to all combine into one. So Brett and I combined a few years ago, maybe a year or 2 ago actually, into the VWM Families Foundation, and now Nairi from Saving Chloe Saxby is winding that down, and she'll be joining our board. So we now have one organization, patient organization, that is the primary point of contact for all the clinical trials and to work with the consortium. So it makes everybody's life a lot easier going forward. So just to say, we've, thanks to you guys, because all of the families, we've raised $490,000 over the last 5 years, almost every bit of which has gone to research. So, and that's pretty awesome. And right now we're very focused on, I'll get back to that. We're very focused on the Guanabens trial, but hopefully going forward, once that, we finish funding that, we can help fund some of the gene therapy and the other initiatives that are happening. And then obviously, the more help we can get from the other families, the more we can together fund, because obviously this organization is not for us, it's for all of our kids. So I want to talk about, we formed a patient advocacy group. Some of you are on it. If anybody is interested in being on it now or in the future, feel free to contact me. We're trying to get a mix of locations and ages and kids in clinical trials and kids not. So, when spots open up, we can fit kids in. The main points, the main purpose of the patient advocacy group is going to be able to provide input to the clinical trials. The Guanabens trial, the Calico trial, the Denali one, if that comes, and to work closely with the consortium for whatever they need. As these clinical trials are coming, they're going to need more and more patient input, especially when we're looking to go to FDA. A lot of patient input is going to be required. So we don't need to do a lot right now, but there is going to get to a point where they will need a lot of information from patients. In the meantime, what we want to do as the PAG is to advocate for the new diagnosed, make sure we're pointing all of you in the direction of the best resources, what's available to you, what clinical trials are available, and how best to manage VWM within your families. And then also something else we're looking to do, which we'd like help with from everybody, is to create protocols and guidance for families. We don't really have something, I feel like we all have stuff in our heads. Everything has been tried by a family somewhere, and it's a matter of just getting it on paper and making sure it's available to all the families so they can see, okay, they tried this, it didn't work for them. And Jenny and I have started working on that, and so as soon as we get a kind of a draft together, we'll send it out to the PAG and get much input as possible, and then we'll be able to get that on the website. So we've got a fundraising committee. Erica has graciously offered to help run it, thank God. and then me and Chelsea also is going to help. We've decided to kind of consolidate into 2 fundraisers a year so that it's super easy for everybody, and hopefully keep doing the same fundraiser so everybody knows there's coming. There's one in April, there's one in September. And as obviously as much participation as we can get, the more money we can raise and the more fun, the more initiatives we can fund. Where we do need help, this is for families, we need help kind of starting to get ourselves a little bit more organized in terms of websites and social media. So we want to create all this content, but we really need help getting it out there. Because, obviously Brett and me and Donna have a lot going on, and there's everybody. So if we could get, the more people we could get to help, the easier it will be. So just contact me if you want to help. Oh, and this is the last thing I have to say. And what we always get asked is most of the PAGs are very focused on newborn screening. Why aren't we focused on newborn screening? We're not focused on newborn screening because it's not feasible for VWM currently with the current technology the way it is. Because VWM is so unique in that it's on one of 5 genes, and each parent has a very specific genetic mutation, and each of them are different, and there's over 160 that have been identified. The only way to test is if you already know what those mutations are. So right now it is not possible to test for it unless you know the mutations. So if somebody has a child with VWM and then decided to have another child, you could test for it, but you cannot test the general population. It's simply, the technology simply isn't there to do that. So we're actually super lucky because it's very expensive and it's very time consuming to get into the newborn screening role. So it means that we're able to focus a lot of our efforts a lot more on research and hopefully finding treatments for the kids who are already here. And that's really all I have to say. Yeah. So about what? Yes, please say something about newborn screening.
Okay. For newborn screening, there needs to be a treatment available. So, that's the first thing, and hopefully that will not take too long. And then the problem is we have 5 genes. So we talked about it, I think yesterday or so, and you would need next generation sequencing techniques. Because there are so many mutations, it's not a simple biochemical test. Like if you screen for ALD, you just have to look for very long chain fatty acids, and you can screen for that. In vanishing white matter, we don't have that. So it's principally feasible, but technology-wise, it's still very demanding. But certainly something that the consortium is already thinking of about how that needs to be, because as soon as there's an effective treatment available, I think that the next day people are on the floor step and asking, okay, can I now have neonatal screening? So we are thinking of it and working on it.
Excellent. All right. I assume nobody has questions. Oh, question. Hi.
For the email for the registry, I'm not sure if the VMC is still...
Oh, I've got 2. Yeah, I wasn't sure which. Did they both work?
Okay, they both work? Oh, so...
Yeah, and I don't think that they will take it out from the air, because there are so many referred to, it just ends in the same inbox.
Okay, then that's perfect. No,
good.
I didn't say anything.
Phew. Because I've been giving everybody the 2 emails, so...
VUMC is shorter, but
Jenny.
One thing. Hi, my name's Jenny. So one question I have for all the researchers in the room is, for those of us that have children that are very progressed, obviously, like we want to find a cure for the disease and treatment, but we really need help figuring out how to take care of our kids now.
Yeah.
And so I would really like to see more research focused on how we can take care of our kids and give them the best outcome possible. So specifically, like, I love the recommendations for keep a fever down, avoid head injuries, but even some more of those general terms, like the oxidative stress, like I don't know what that is, you know, and what causes oxidative stress, and what are different things that we can give to help with that, and ER stress. I don't know what that translates to in real life. So I would really like to know a full-on list, like what causes stress, that affects the disease. Even like heme deficiency, which I know is like an iron deficiency, or dehydration, and we just need like better parameters.
But I'd also love to see, a lot of the medications that are prescribed. I've noticed that my son has very severe reactions to medications very often, and I think that's pretty common. It's like it would be nice that these medications that are prescribed for everything, could we test them on the mouse models and see if they have an effect? Because personally, I've seen effects. I've given him a drug and his speech is slowed immediately, and I take him off it. But I feel like there's a lot of treatments that our kids get that aren't necessarily great for our kids.
Okay. Last Tuesday, we had a consortium meeting here. So everybody was a day early, and we discussed, the consortium was founded in 2019, so it's 4 years. And then after 4 years, you really have to sit and say, okay, what have we done, and what are we going to do? And we discussed this point that we also need to contribute to writing recommendations and information for families. And also, we discussed this, a letter that you can take to the ER when your child is sick, so that you did put together. You did put together, and I think Donna. We put it on the Amsterdam website, so that and Amsterdam stationery, so that it has some authority. But as a consortium, we also recognize the point, and that's also on our to-do list for the coming 4 years. So it's joint effort, I think. Yeah.
Hello, everybody. Good afternoon. My name is Bill Cho. I am a clinical research physician at a California-based biotech company called Calico Life Sciences. And I'd like to thank Mario for giving me this opportunity to share with this audience some of the progress that we've made on a drug development program for a molecule that we call ABBV CLS7262. It's got this crazy convoluted name because it's not really a drug yet, so we still have a code name for the molecule. This molecule is in a class of molecules called EIF2B activators that you heard earlier about, and we're hoping that this drug development program could lead to a potential treatment for people with vanishing white matter disease. So I was going to give you some highlights of our drug development program, but before going there, I just wanted to say a few words about who we are, Calico, since we're a fairly small company, about a couple hundred employees. So maybe many of you have not heard about us, but we're based in the San Francisco Bay Area. We were founded about 10 years ago by Google, and they're now known as Alphabet. And Art Levinson, who is our CEO, and Art Levinson was the former CEO of Genentech before he founded Calico. And we have a fairly ambitious mission, and it's a unique mission, I think, among drug companies, and that is to try to understand the biology of human aging and to develop therapies for age-related disorders, including treating disorders that affect the brain. And since we started in 2013, we've been in a collaboration with a large biopharmaceutical company that you probably all have heard of. This is AbbVie, and they have a long track record for developing drugs for various brain disorders, including Parkinson's disease, schizophrenia, and depression. So together we've been trying to understand human aging, and you can imagine that in this pursuit we start to get ourselves researching some very fundamental biological pathways. And one of the pathways that we became interested in is a pathway called the integrated stress response pathway. And this pathway may be involved with human aging, but we've come to realize that this is very important for the cause of vanishing white matter disease. And so we have a drug development program ongoing. So we developed a short informational video that I'd like to share with you. It's just a couple of minutes, and I thought that would be a good introduction to the talk today. All right, so hopefully that was informative. And I'd like to then take you through some of the highlights of our drug development program. And so maybe I'll start off with reminding you of what the integrated stress response pathway is. It's a pathway that allows all the cells in the human body to react to various types of biological stress. And in situations of stress, normal protein synthesis is decreased and stress protein synthesis is increased. All right, so the two key players in this pathway are enzymes called EIF2 and EIF2B, and I think you've heard a lot about EIF2B already. These two enzymes work together to form the normal set of proteins that cells need to carry out their normal function. But in vanishing white matter, the genes that encode EIF2B have variations in them, and that leads to a loss of function of EIF2B. And in that situation, normal protein synthesis is decreased, and there's an increase in these stress proteins that are depicted by the red squiggles here. And we believe that when the ISR is chronically turned on, these stress proteins can accumulate, they can be harmful to the cells, and that could lead to a loss of function of those cells that express those stress proteins for a long period of time. You can imagine why we and others have been interested in trying to find molecules that will activate EIF2B. And the first major breakthrough came from the discovery of this molecule called ISRIB, or ISRIB, which stands for ISR inhibitor. And this molecule was discovered by a scientist named Carmela Sidrowski. She was a research scientist at the University of California, San Francisco, when she discovered this molecule. And you can see the image of the molecule up here. Carmela now works at Calico, and she's continued her research in the ISR at Calico. And so the way this molecule works is it binds to the center of this complex EIF2B enzymes. You can see how the molecule sort of is symmetrical and have these two different arms. And what this does is it stabilizes the formation of this complex, increasing or boosting the activity of EIF2B. And so when EIF2 encounters boosted activity of EIF2B, then normal protein synthesis can restore back to normal, and then we can decrease the amount of stress proteins that are found in vanishing white matter. So we've now made several iterations of ISRIB that improve on the chemical properties, and we started testing them in mouse models of vanishing white matter. And I think you've heard about some of these mouse models earlier. We created a mouse that has the R191H mutation in the 5th subunit, and this mouse has a lot of the same features as clinical vanishing white matter disease. So they lose the white matter in the brain and the spinal cord, and they also have some problems with their movements and coordination. And so we tested these 2B activators in this mouse model, and here these are microscope images through the spinal cord of this mouse. And on the left, you can see that the white matter here has been stained blue, and you can see that in the normal mouse there's a lot of blue staining there, which indicates a normal amount of white matter. And then in the vanishing white matter mouse, this blue staining is significantly decreased, showing this loss of white matter. And when these mice are treated with 2B activator compounds, we can prevent the loss of this white matter in this mouse model. We also wanted to test whether or not these 2B activators would have an impact on the function of these mice. And so you've heard about a similar assay before, the catwalk. This is a balance beam assay where the mice are put on a narrow balance beam and they have to walk across, and we time how long it takes these mice to cross. And so on the vertical axis here, we have the time to cross the balance beam, and on the x-axis, the horizontal axis, we have the age of the mouse during the experiment. And we did this experiment between the ages of 24 and 35 weeks of age, which is an adult mouse. Normal mice can cross this balance beam fairly quickly, in about 5 seconds, and that's shown by the black line here at the bottom. This is what a normal mouse looks like. At 24 weeks of age, they can cross in about 5 seconds, and as they get older, they're still able to cross in about 5 seconds. But if you compare that to the vanishing white matter mouse, starting at about 24 weeks of age, you can see that it takes them a little bit longer to cross the balance beam, probably because of coordination problems. And then as they get older, that problem gets worse, and it takes 30 seconds to cross the balance beam towards the end of this experiment. But when these mice are treated with 2B activator compounds, you can see that there is a stabilization in the worsening of their ability to cross. And it looks like maybe at the very end there might be a slight improvement in their ability to cross the balance beam. So we were very encouraged about these results in a mouse, but of course the mouse is not a human. So we really felt that it was important to start clinical trials with these molecules. So we picked among these 2B activator molecules the one that we wanted to take into the clinic that has the best drug-like properties, and we call this ABBV CLS7262. So we started a clinical development program a couple of years ago, and you can't just jump straight to patients with a brand new molecule. So what we and others typically do in the pharmaceutical industry is to start trials in healthy volunteers, normal, healthy adult subjects, to see how the drug behaves inside the body and to try to understand the potential side effects of these new molecules. So we conducted phase 1, we call them first-in-human trials, and in these trials we've now given this molecule to over 100 healthy subjects. And what we learned in these studies is that this molecule can be administered orally, by mouth, once a day. The side effects were non-serious and they were mild to moderate in severity. We extracted the blood cells from these healthy subjects and we measured EIF2B activity in the blood cells, and we showed that we can boost EIF2B activity in these blood cells and we can also suppress the integrated stress response as expected. Earlier it was mentioned that there is a blood-brain barrier, and there is a blood-brain barrier that keeps foreign chemicals outside the brain. And so you really have to make sure that the drug crosses the blood-brain barrier and get into the brain. So we did spinal taps on our healthy subjects, and we measured the amount of drug that gets into the cerebral spinal fluid, which is the fluid that surrounds the brain, and we measured the concentration of drug, and we believe that the concentrations in the cerebral spinal fluid are high enough to impact EIF2B in the brain. So the next step was to start testing this molecule in patients with neurological disease. And I didn't mention this at the beginning, but we're also interested in developing this molecule for ALS, or motor neuron disease. And so we conducted a small phase 1B study in people with ALS. And this is a fairly complex study design. There's two parts to it. There's a part 1, which is a 1-month-long study period where subjects get randomly assigned to 3 different dose levels of 7262 or placebo, which is a sugar pill that looks like drug. And then everybody gets to go into the second phase of the study where everybody gets active treatment extension for several years. And this study is still ongoing, so we don't have all the data yet, but at least the early enrollees in this trial have been in this study for about a year. And so we can start to understand some of the long-term side effects in people with ALS. So these are the side effects that we've tabulated so far during this 1-year study. And this drug has been given to 31 people with ALS, and these are the most common side effects that have been observed. They're headache, nausea, itchiness, constipation, dizziness. These are very similar to the side effects that we saw in healthy volunteers. So we were very encouraged by the safety profile in healthy subjects and people with ALS, and so we thought we were ready to embark on a drug development program in vanishing white matter. So we have, I'm happy to announce that we have kicked off a small phase 1B/phase 2 clinical trial evaluating 7262. And this is the first study with this molecule in people with vanishing white matter disease. It's a very small study that's just gotten started in April, but we're hoping that this will be part of a much larger vanishing white matter drug development program depending on the results of this study. So the purpose of this small initial study is to look for side effects in people with vanishing white matter and to understand the drug-like properties in this particular population. Currently, we're only enrolling adults, and we have one site open at the University of Utah, and the doctor that's running the trial there is Dr. Josh Bonkowski, who is going to be speaking next. But we're currently amending the trial to expand it to include adolescents and children with vanishing white matter, a small number of children. And we plan on opening a second center in the Netherlands at VWM, and Professor Dr. van der Nap will be the doctor running the trial at that site. And so, as I mentioned before, we're hoping that the results from this study will be positive, and these data will be helpful to inform future clinical trials in vanishing white matter. So thank you for your attention, and thank you to the patients who may have participated in our phase 1B trial to date. I'm not sure if anyone's here or dialed in on Zoom, but thank you for your participation.
So I apologize that I can't be in there in person. I seem to have overcommitted myself as usual, but I'm there with you in spirit. And so I'm delighted to have a chance to talk with everybody. And really, at this point, I think you've heard all the key things. And I was just going to briefly talk about kind of the first steps that we've taken in this phase 1B clinical trial. So this is a huge collaborative effort, including myself at Primary Children's Hospital in the University of Utah, in Salt Lake City, Utah, Dr. Van der Knapp in Amsterdam, and the Calico team. So it's exciting to be up here. So just, the study is funded through by Calico, and there's various disclosures not really relevant directly to the study, but just as a point of specificity to the work. So I thought I'd briefly mention that when we think about a clinical trial, the whole point is that we don't really know for sure if it's going to work or not. Of course, we have high hopes, and we would be very delighted to see this drug or any treatment work for vanishing white matter disease. And as we go to bring a treatment forward, there's a whole bunch of different steps, and there's different phases of research. I'll try again. So there's different phases of research, and those different phases are kind of important to understand for how the study works. So the first step that Dr. Cho was talking about was a lot of work that had to be done in the lab first, both in chemistry and then in mice at the bench, and that's kind of what's called T0 research. So making sure we understand the disease, and it also was work from a bunch of labs, including Dr. Van der Knapp's, to understand how vanishing white matter disease causes disease. Now we're kind of trying to move into clinical trials. So, and there's different phases of clinical trials. So this is kind of the T1 and T2 steps with different stages of clinical trials. I'll talk about that more in a minute. And that's supposed to kind of make sure the drug is safe, understand what doses work, and then eventually kind of the T2 phase, do a more advanced clinical trials to understand, like, is it really effective in stopping disease or changing disease course? And then, of course, at some point, we want to bring it out broadly, and that's using it clinically. So with FDA approval in the United States or EMA approval in Europe, and then in T4, kind of broad use of the drug. So all these steps have to kind of occur to really make a difference in the disease. So in the clinical trial, there's these different phases everybody might have heard of, and they're complicated, and I apologize, this is kind of the way it is. So phase 1, and the first step is just understanding, is the drug safe to use in a person? Do we understand what dose we should use? Because if you're not giving enough, then you can't hope to see an effect, or if you're giving too much, maybe you're just exposing people to potential side effects. We want to make sure if there are any side effects that they're not risky or they're very minimal. And so that's kind of a first phase. Phase 2 and phase 3 are where, again, looking at safety, looking at side effects, and really testing effectiveness. And then phase 4 kind of is oftentimes post-approval and kind of further follow-up of the drug once it's being used in clinical practice. But usually after phase 3 or phase 2/3, there's enough information that the FDA or other health authorities would approve a drug. So really, phase 2 and phase 3 are critical for getting that approval for the drug to be available to everybody. That's the simple. So I feel like each slide gets slightly more complicated, right? And so now we're talking about like different stages that are more complex. So there's something called like a phase 1B, where you're still in a phase 1 step as part of the study where you're looking at the dosing, but you're looking at different types, what are called like ascending doses. So starting with a low dose and moving to a slightly higher dose. And so depending on how many subjects you have and on how big the effect you can see, you kind of have to choose how many people you can do this in. And so the number of subjects is kind of a broad range. And so prevention of white matter disease, because it's a much rarer disease, and we just have, we're hoping we're seeing a stronger effect. All these numbers here are going to be much smaller. And so the study that we've started in Utah with the phase 1B, and then that's expanding now with ourselves and with Amsterdam, will be a phase 1B/2 study. And so what are all the parts that we're involved in? And so I apologize because we're doing the study kind of, it's live. We're underway because of the confidentiality and not kind of affecting the results of the study. I can kind of only speak in some generalities. And so I'm not trying to be vague because I want to, but because we have to be deliberately vague just to kind of keep confidentiality about the study and not influence or bias ourselves for the study. I'll describe what I can. So what's involved in the study? And so when we want to bring somebody in for the study, we have to do much of preparation, contacting the family and the patient, making sure we can arrange dates and the clinic site for their visit. At the initial visit, we're doing physical and neurologic exams. There's blood draws and a lumbar puncture, or what's called a spinal tap, involved, and an MRI to have a kind of a baseline MRI. We give the drug, and then there's more blood draws because we have to understand what the initial response in the body is to the blood. Then there's a series of subsequent visits over the next days and weeks and months to look at how the person's doing, make sure there's no adverse effects, and again to do more blood draws to look at the levels of the drug in the blood and make sure that it's at a level that we think is going to be effective. Then depending how the person's doing, with no side effects, we adjust the dose, and we have kind of a plan for how we're going to adjust the dose if everything's going okay. And that's usually in the first 2 to 3 months that we're doing this. And again, we're not testing effectiveness. We're not adjusting the dose based on effectiveness. We're basically doing it based on safety and based on the lack of there not being any side effects. And then there's kind of a timeline where we do blood draws, visits, another MRI, one more lumbar puncture, and then the whole kind of first part of the study runs over 2 years. So it's quite complex. Like I've boiled it down to a very kind of condensed 1-slide picture, but it's been a huge process. Like we have about 3 or 4 what are called study coordinators at the University of Utah working on this study almost full time. We've worked with multiple people from the Calico team because they're trying to make sure we have all the processes in place. And then also what's called a clinical trial organization that helps kind of run the day-to-day logistics of the study. So there's a bunch of regulatory paperwork and legal processes that have to be met because you don't want to do this in sort of an offhand fashion and then have the FDA kind of get hung up on a technicality. And so that's why there's so much complexity to it. It is a huge time commitment from the patients and families. And so, they in a way are really helping to make this drug available by putting their time forward for the study. And then the study teams are also doing a lot of work to make this all possible. So how's it going? So far so good. And again, sorry for the deliberate vagueness, but so everything's going great. We're happy, and I think the Calico team is happy too. They seem smiley when I see them, so that's usually a good sign that the study is going well. So we've had to work through a lot of processes. So in a way, we've worked out some of the bugs for how the study goes. And I think, although it's been somewhat annoying and frustrating, on the other hand, I think we understand the points of confusion and know what to do better. Kind of interestingly, the drug looks different than I thought it would. It kind of looks like Pop Rocks. It doesn't taste like that, but it kind of looks like Pop Rocks, if everyone knows what Pop Rocks looks like. And then I think, understandably, no one's excited about the lumbar puncture. I think there's a lot of anxiety on patients and families' part, but with the sedation, it's just been totally fine. And so I want to reassure people that, although it's a little bit a hassle and a little scary to think about, that the process has gone super well. So just to try and reassure people that parts ended up being not a problem. So just to kind of summarize, the phase 1B is underway, and then we're looking forward to the next few months to get the phase 1b/2 part of the study expanded. The purpose of this phase 1b is to test the dosing, to look at drug levels, to make sure there's not any unexpected side effects as we use it in people who actually have vanishing white matter disease. It's a huge collaborative effort, so we're really indebted to the work of Mario, to the Calico team, and to the Vanishing White Matter Disease Consortium, which has really been instrumental in helping to drive this process forward. The patients and families, the people who are participating, as well as the entire kind of community, has been integral in helping move this forward. So it's hard to express enough kind of my thanks for everybody for being involved. And we do have a great study team. So if you see members of the study team floating around from Utah, please say hi to them and thank them. There are some caveats to the study. It is intentionally small because we're trying to keep it as small as possible for these first steps, just to make sure we know what we're doing before we can go large. And we're trying to go as fast as we can on the one hand, but we're also trying to be as safe as we can at the same time. And so it's a delicate balancing act to get it all right. So thanks for your attention. And that was kind of a brief and vague introduction to the phase 1b study, but it's exciting that we're here and this is moving forward.